We're going to take a look at the scientific method. In order to test the hypothesis that we've come up with, a lot of scientists will do the scientific method, which is just a series of steps that are used to go through and do our experiments. But the scientific method is not set in stone, and sometimes these steps will be kind of put in some different orders, or we'll kind of come back and repeat some steps. And there are other methods besides the scientific method which are used to test hypotheses as well. So for example, I'm in a scientific study called the All of Us Research Program, where they've taken my DNA samples and measurements and blood samples and have access to my medical records. And it's a big nationwide study, and what they're doing is they're looking at DNA and genes and also looking at people's medical records to see what kind of diseases they have, and trying to see what genes are associated with what diseases, or what diseases people might develop. So it's kind of looking backwards instead of actually doing actual experiments, because things have already happened. So there are different ways of doing science. But let's just take a look at the scientific method. So we start with making an observation about something in nature, and then we ask a question about that. We propose a hypothesis, which is just a tentative answer to our question or explanation of that observation. And then we make some predictions, which we can test, and then we do experiments to test the predictions. And these experiments must be repeatable by other scientists. And then we'll analyze the results and then draw conclusions. So either the hypothesis will be supported by the data we've collected, or it's not supported, and then we reject our hypothesis and then we start over. And then we report the results in peer-reviewed journals. So the journals that these scientific papers are published in are actually reviewed by scientific peers who are also experts in that field, and they kind of read through things and go through things with a fine-tooth comb and make sure that everything looks good before it's published. Okay, so let's take a look at variables. So a variable is just anything that can change or vary in an experiment. And there's three different types of variables. So an independent variable is the factor that's going to be changed by the researchers. So usually you just pick one thing that you want to study in your experiment, and that's what you're going to be manipulating and changing between the experimental groups, and that'll be your independent variable. And when you're graphing things, the independent variable is going to be put on the x-axis. And then the dependent variable is the factor that we're going to be measuring that's predicted to be affected by the independent variable. So it's called the dependent variable because it depends on the independent variable. So it's going to be affected by that independent variable. And on the graph, it's going to be on the y-axis. And then we also have these constant, sometimes called controlled variables, and these are going to be the factors that stay the same in all of our experimental groups. And this helps keep the experiment controlled, because you don't want too many differences between your experimental and control groups. So for example, if you're doing experiments with people, and you're testing like a new drug, you want to make sure, maybe, diets are the same between the two groups, or maybe you want to make sure the amount of sleep they're getting is the same. There's a lot of different factors that, you might want to keep the same between the two groups. Okay, so here's just a nice picture that just shows 3 different types of variables. So here we're taking a look at our independent variable, the thing that you're going to change. So what we're looking at here is the amount of water that we're going to be giving these plants. So that'll be our independent variable. So that's what we're going to change between the different groups, how much water we're giving the plants. And then our dependent variable that we're going to be measuring would be the plant growth. So that would be our dependent variable that's going to depend on how much water, that independent variable, that will change the amount of plant growth. And then these controlled or constant variables, in this example here, maybe it's the type of soil, the pot size. We'd want to keep that the same between the different experimental groups. Okay, so after our experiments that we've done, we've got different groups of plants that we've given different amounts of water to. And here we plotted in different colors the amount of water. And then here on our y-axis is our height of the plants. And then on the x-axis here is the amount of water. So which one is going to be our independent variable? Remember, that's the variable that the researcher has changed. So they've divided the plants into different groups and given each plant a different amount of water. So our independent variable would be here on our x-axis, and that's the different amounts of water that each group of plant got. And then our dependent variable is going to be affected by the differing amounts of water, and that's going to be on the y-axis, and that'll be the height of the plant, how tall that the different plants got. Okay, and a lot of times we're going to be using these controlled experiments to test our hypothesis. And what this basically is we're just going to run 2 experiments in parallel. So we'll have an experimental group and a control group. And the only difference between these 2 groups is that the experimental group, we're going to change the independent variable to see what its effect will have on that experimental group. And then in the control group, the independent variable will not change. So for example, maybe we want to take a look at how camouflage will affect how much these mice will be preyed on. So our control group, we're going to leave the mice camouflaged, but in our experimental group, we're going to change their color. Okay, so we're going to make them non-camouflaged. So our independent variable would be whether they're camouflaged or not. The control group, we're going to leave them camouflaged, but our experimental group, we're going to change the color and make them so they're not camouflaged, so they stand out. Okay, so let's just give you an example of the scientific method. So let's say that our observation, we observe that we've got same species of mice, but they have different coat color patterns and they live in different environments. So we've got a population of mice living on the beach and they're white, but the same species of mice also lives inland and they're brown. Okay, so the same species of mice, but depending on where they live, they're either white or brown. So we ask a question and say, well, what's the function of camouflage? Okay, because the white ones blend in really nicely with the white sand, and the brown ones blend in pretty nicely with the inland where you've got kind of this brown vegetation. Okay, so we come up with our hypothesis, our tentative answer or explanation for our question here. And our hypothesis is that coat coloration Camouflage is an evolutionary adaptation that decreases the mouse's risk of being eaten by predators. Okay, so now we come up with our prediction. So we predict that mice with the non-camouflaged coat coloration would be preyed on more heavily than mice with the camouflaged coat coloration. Okay, so with our experiment, we're going to have a control group and an experimental group. So the control group, we're going to leave camouflage. That's their normal way that they are. But the experimental group, we're going to change that independent variable. Okay, so this is the independent variable is what the researcher's going to manipulate, and we're going to change the coat color and make them so they're not camouflaged. Okay, so they will stand out. Okay, so here we've got a beach habitat and our inland habitat here. And what they did is they actually made these mouse models out of kind of a plastic kind of clay model stuff. And then they set them out, left them out for about a week, and then they came back and counted the bite marks on these kind of clay plasticized models. All right, so let's take a look at these graphs and which one's going to be our independent variable. So here's we've got our x-axis on the bottom and our y-axis over here. So x-axis we've got the color, right? Okay, so whether they're camouflaged or not, so whether they're light or dark, and then that would be our independent variable, which is on the x-axis, whether they're camouflaged or not, the color. And then our dependent variable is going to be on the y-axis, and this would be the number of bite marks, so the percentage that were attacked. Okay. So you can see in the beach habitat, the ones that were light colored were not as attacked as much as the one that were dark colored and stood out. Okay, and then in the inland habitat, the ones that were dark didn't get as attacked as much as the ones that were light colored. Okay, the ones that were light colored got more bite marks on them. Okay, so those were our results. And then our conclusion is that our hypothesis was supported. So the mice with the camouflage coloration were preyed on less often than the non-camouflaged mice. Okay, what if you're doing experiments on humans? When you're working with humans, we have the possibility of bias. So bias from the participants, and then also bias from the researchers. So we want to try to eliminate bias. So the ways that we eliminate bias would be randomization and double blinding. And both of these are going to help decrease what's called the placebo effect. The placebo effect is when the participant is taking the placebo, maybe like a sugar pill or something, and they think it's actually the medication, and their brain tricks them into thinking that they're getting better when it's really not the treatment. And so that can interfere with the results of the experiment. So by doing this randomization and double blinding, that can get rid of the placebo effect. Okay, so randomization is just when the participants come into the trial, they're just going to be assigned randomly to either the control group or the experimental group. Okay, so it's just random which one that they get assigned to. And then double blinding means that both the participant and the researcher are not going to know whether the participant is in the control group or the experimental group. Okay, so nobody knows which group the participant is in. Okay, because the control group is going to get the placebo and the experimental group is going to get the treatment. Okay, maybe it's the medication or whatever. So both groups, look like they're getting the treatment because the placebo will look like the medication, but it's not the medication. Okay, and so both groups look the same, but one is getting the placebo and then the other one is getting the real medication. Okay, so for example, I did a clinical trial where they were testing a medication to help with chronic fatigue syndrome, which I have. And I'm not sure which group I was in, so they never told me. And they just randomized me into one group or the other. And the researcher didn't know which group I was in either. And they gave me a bottle, and it contained either pills with rice powder or, the actual medication they were testing. And I didn't know which one it was. Okay. And they never actually even told me, so I had no idea. And that was, double blinding and also randomization at the same time. So this helps get rid of this placebo effect. Okay. They also try to make the groups as similar as possible. As far as, age ranges and genders, and things like that too. So they try to match the groups, as closely as they can, just to try to, rule out, effects of gender and age and things like that as well. So